Hello and welcome everybody to the webinar this evening on the roles of lymphatic fluid flow in immune system function with Professor James Moore. Really excited to have the webinar, second webinar with Professor Moore, or also known as Jimmy, with us at PhysiQuip as a follow-up to the one that we did on the effects of positive and negative pressure on lymphatic pumping. So we have a really good turnout on the webinar this evening. We have around about 100 people who registered. We're expecting about 60 people live. So hopefully we'll get plenty of interaction. So for those who are joining live, thank you very much for taking time out this evening and joining us. The webinar is going to be split into two parts. Jimmy will share with us a presentation and then we'll have some time at the end for some Q&A. So if you do have any questions that pop up, we can get them answered for you at the end. Now the session is being recorded, so if you do miss anything or you want to revisit any of the content, you'll get access to this via a link that we send out in an email at the end. Any questions that you do have during the live session, there's a Q&A function at the bottom of the screen. Type your question there and we'll get it answered. For anybody who is watching on the recording, if you have any questions that you'd like to send in, our email is at info@physiquip.com and we'll get it answered for you. So very quick introduction to myself. I'm Dominic Smith, Clinical Applications Specialist at PhysiQuip. For those of you who aren't aware of PhysiQuip, we are a UK-based medical distribution company who work with several international partners. Our aim is to bring some of the leading technology from around the world into the UK market. Not only is it technology, it's also some of the world's leading speakers and clinicians to our education platform, hence why we have Professor Moore with us this evening. So with that introduction to us, I would like to hand over to Professor James Moore Jr., also known as Jimmy. He joins us from Imperial College London, albeit this evening from the Home Office. Jimmy, I'll hand over to you and you can get going with your presentation.
Right. Thank you, Dom, and thank you, Katie, and the whole team at PhysiQuip. You guys are great. We love what you do, and are glad to have you in the community. And it's a great honor to be invited to give a second seminar with you. It's I really enjoyed the first one, got some great questions in, and I expect nothing less this time. So so with that, I'll go ahead and introduce the team that's really involved here. This is me in the back there, the old guy, and my postdocs and PhD students, and one of their furry friends. And so the work you're going to see today was really done by these people and our collaborators. And just want to really make sure I acknowledge the right people in that sense. So we have a lot of projects in the lab that are listed on the left there. You heard about some of the pumping analysis last time. This time we're going to be talking more about the immune system. But in all cases in our research, we do some experiments, we do some computational modeling, and we do some device development. So you're going to hear mainly about the experiments in modeling today, so not so much about the device development. But the idea is that what we're after is a better understanding of this very complex system. And really the best way to understand a complex system, in my view, is to approach it from all angles that you have available to you. So what's the best tool or combination of tools to understand that system? And for us, that almost always involves some lab work where we get our hands dirty, and then some computational work as well. So with the lymphatic system, I always like to give a bit of an introduction because it is a bit of an obscure topic medically. There are not many people that study the lymphatic system, maybe a few hundred biologists scattered around the world. So its main job is fluid balance, and that's what we talked about last time. It pumps fluid from the peripheral tissues back centrally to the venous system. And because it acts somewhat like a sewer system, it has to clean all of the fluid that goes back, and that's the job of the lymph nodes. And we're going to spend a fair amount of time talking about lymph nodes today. So the lymph nodes are important for adaptive immunity, the major topic for today, but also for cancer metastases, and that's a huge medical problem, obviously. So in terms of raw numbers, the blood flow is pretty high volume, high pressure, and fairly large vessels. In comparison, the lymphatic system only pumps a small amount of fluid around the body. every day. It does so at very low pressures and indeed at sub-atmospheric pressures. And the vessels are pretty tiny. So the largest vessel is about 2 or 3 millimeters in diameter. Most lymphatic vessels in your body are under 1 millimeter in diameter. So quite tiny vessels with a mostly transparent fluid going through them. They're easy to miss for surgeons cutting through. So it's I think that's one of the reasons it's not been studied as much. So we're going to talk a bit today about adaptive immunity, and it's important, particularly in the experience we've all had in the past year here with COVID, to talk about that in the framework of vaccines. So the idea of vaccines is to get your body to recognize things that might harm you in a way that's safe, so that you can develop immunity to that particular antigen or that thing that might harm you. So the real goal is to get the immune cells to recognize and respond to the antigen. So the antigen in this case is injected with a syringe into the muscle in your shoulder. I'm going for my second injection on Saturday. So in about 48 hours, I'll be getting that in my arm. And so that goes into the muscle. There are some lymphatic vessels in muscle, so the antigen information can just go directly into those vessels, and then it gets pumped into the lymph node. And the lymph node is where you develop adaptive immunity. So the goal is to pass that information from the syringe to the B cells and the T cells. So these are important immune cells inside the lymph node. And the lymph node is just chock full of these cells. So these B cells tend to be in little clusters around the edge, and then the T cells will occupy this whole spongy region in the middle here. And I'll show you some other pictures of that later. But that's the real job, is to get that information from the syringe to those B cells and T cells, the lymphocytes that are inside the lymph node. And as I said, the lymph node is chock full of these cells. They're at really, really high concentrations. The B cells and T cells, you find just millions of them inside a typical lymph node. Compared to, say, the skin or the muscle, you don't find that many lymphocytes, these T cells and B cells. So if you compare the population density of T cells and B cells in the lymph node, it's something like New York City. So the population density of New York City is really high. And if you draw an analogy to the number of T cells that you find in a lymph node, then the number of T cells you would find in muscle would be, it would have the population density equivalent to about that of the UK. So if you want to pass information on to a large number of T cells, and you need to get in close contact with those T cells, then the best place to go is inside the lymph node, where they're densely packed. So this is what it looks like when antigen comes into a lymph node. So this is not from my lab. This is from Mike Carroll's lab, who's at Harvard University in the States. And so what he's done is labeled a small antigen as red here. And you can see that comes into the lymph node, and it goes down these tiny little passageways called conduits into the deep spongy part of the lymph node. And in this case, the B cells are what you see in white and in blue. And you can see that as soon as the antigen shows up, these cells take up the antigen, and then they start telling each other about it. So if you pick a little white cell here and follow it around, you can see it's going around and talking to its neighbors, and then it goes and talks to somebody else, and it goes to talk to somebody else. So it's like a singles bar for immune cells. So this is where they all learn from each other about this antigen, and they're crawling around here at fairly low velocities, but they're packed so tightly with each other that they are able to pass on that information. This is absolutely crucial for adaptive immunity. So if you get a vaccine, once that information shows up into the lymph node, then this is what's happening, as those cells are passing that information around. Now, here is a sort of cartoon representation of a lymph node. So you have these afferent vessels bringing the flow in. You have typically a single efferent vessel taking the lymph flow out, and then there are blood vessels stacked in here as well. So I'll come back to those later. But that's where those B cells are. This is where the T cells are. this region labeled in green here is known as the subcapsular sinus. It's a relatively open region. So this would be the B cell or T cell region here. You can see it's a very tightly packed sponge compared to the relatively open space of the subcapsular sinus. So this flow comes in here and then seeps around the edges, all around the top part of this lymph node here, before the fluid dives into the tight spongy region here. So it spreads around that space and then dives in. And it eventually gets collected in what's known as the medullary sinus and then leaves the vessel. So the flow chart literally looks like this. So you got an afferent vessel, the flow comes in, the flow can go into the subcapsular sinus and around, or it can go from the subcapsular sinus through the spongy part. And so we got interested in what determines the split of the flow from kind of it going around the periphery versus it going around the middle. And that's actually a very difficult thing to measure. So the image I showed you in the previous slide of what goes on inside the lymph node, that's okay for observing cells, but for observing the fluid flow through there, it's not very good. So the best way to figure this out would be to construct a computational model. So that's what we did. And what we find is that most of the flow actually goes around the edge here. So remember that open channel region, the subcapsular sinus, is a fairly low resistance pathway. So most of the flow that comes into the lymph node actually goes around the edge and out the exit, and only about 10% goes into where the B cells and T cells are. So, but it's important to keep in mind, this is under normal conditions, right? If you're not responding to any antigen, you only need to sample a little bit of the fluid that's coming in just to make sure everything's okay. 90% of it can go around the edge here and continue onward. And so the worst thing the thing you don't want to happen is for fluid to start backing up here. And because that causes edema and swelling of tissue. So you want to keep the fluid moving. So this is a pretty intelligently designed little chemical engineering factory that shunts most of the fluid around when there's nothing to worry about. But some of it does come into the B and T cell region here. And so we got more interested in that and wanted to understand how that affects the processes that we talked about. So here's back to this same slide here with the vaccine coming into the muscle, going into the lymphatics, coming into the lymph node. And you may have noticed I mentioned not just the antigen, but the adjuvant here. So what is an adjuvant? Well, it's a general medical term that means kicks up something or makes it better. And so the history of vaccine adjuvants is actually pretty interesting. So it starts about 100 years ago in London, actually in South London. So it was a man named Alexander Glennie who was doing research on vaccines. And he noticed in his animals that if they showed signs of inflammation around the injection site, they actually took up the vaccine better. So then he said, all right, well, let's try and find something that produces a little inflammation and inject it along with the vaccine. And I don't know if this is true or not. This is kind of legend that he looked up on his cabinet and started alphabetically. And the first thing he found was something that started with A, called alum. It's aluminum salt. And he injected that and it seemed to work okay. And literally it took off from there, and it's been used in millions of vaccines around the world. We all have had vaccines, guarantee you, with aluminum salts in them. It continues to be the most common vaccine adjuvant out there. The problem is we don't know how it works. We don't know why it works. So we don't really know. And Glennie himself, back in 1925, just kind of poking these animals and seeing what happened, proposed something called the depot theory, which is that if the aluminum salt creates inflammation at the injection site, attracts immune cells to the injection site, and therefore they take up the information. And he didn't really say much about lymph nodes. He didn't know what we know now about the density of cells in lymph node, kind of that New York City comparison that I showed you earlier. But that's the depot theory is the dogma, and there's not much experimental support for it, but 100 years later, people still believe it. There are immunologists that swear by it. Actually, we don't know. There's not that much known about the way adjuvants work. So since then, there have been other kinds of adjuvants developed. So oil and water emulsions and it's biological nerdery, but TLR activators, toll-like receptors. So no one knows how really they boost immunity either. So if you're a vaccine designer, how do you pick between aluminum salts or oil and water emulsions or TLR activators? It's really almost a random choice. It's also worth pointing out that around the same time in the 1920s, Glennie was doing this. There was a Frenchman who discovered the same thing. His name was Gaston Ramon, and he was trying to induce inflammation by injecting breadcrumbs. So there you go. Well, I guess we're glad that aluminum salts took off and breadcrumbs didn't. But it's an interesting story in science for sure. Now, one thing that we all know about lymph nodes is that they swell when we get ill. So this is an experiment that we can do in mice. So we can inject one of these vaccine adjuvants into a mouse's foot and then dig out the lymph node that is closest to that foot. And what we will see is that this is a normal lymph node over here, kind of at 4 different slices. And then this is the lymph node with the TLR activator, the vaccine adjuvant. And you can see the lymph node is larger. These are measurements of the weight of the lymph node 24 hours after the injection, 72 hours in the injection. You can see with the adjuvant, these lymph nodes definitely get bigger. And that's something we all know, that when we get ill, the lymph nodes, say around our neck, will swell up. It's also a sign of cancer. Potential cancer spread as well. So don't ignore swollen lymph nodes. So we can do this, but then we wanted to know what effect that has on those flow patterns that I mentioned a moment ago. And so we did this experiment where we wanted to measure flow resistance of these lymph nodes. So I thought that it's worth explaining what flow resistance is. So if you look at the diagram on the left here, if you imagine a bucket of water, and then we've got 2 tubes leading out of it. One of them is a big tube, the other one's a small diameter tube. And so you can imagine a lot more of the water is going to go out the big tube here. So that's a low resistance pathway. And a high resistance pathway would be that small tube where less flow is going to come out. So we want to measure the flow resistance of these lymph nodes following vaccination. So this is what it looks like. Mouse lymph nodes are about 1 millimeter in diameter. Our lymph nodes are about 1 centimeter in diameter. So these are tiny little lymph nodes, but we can work with them. And so what we do is we cannulate the afferent, or the incoming vessel here. We shove a small tube inside of it, and we cannulate the efferent vessel, or the outgoing vessel. And we measure the flow rate across there. So how much flow is coming out of this tube, and then what the pressure difference is. So how high is this bucket of water? And one thing I'd like to point out here is these vessels are tiny. I mean, the tubes that we use to cannulate these vessels are on the order of 50 to 70 microns. That's about half the diameter of a human hair, right? So these are exquisite little measurements and tiny, tiny flow rates. So it's a fascinating experiment, and the people that designed the equipment for this are really, really smart, and the people that use it have very steady hands. So this is what happens to flow resistance. So this is flow resistance on this axis here. A healthy lymph node has a relatively low flow resistance because most of that flow is going around the edge, as I showed you earlier. 24 hours after the vaccination, the lymph node flow resistance goes up by an order of magnitude. So it takes a lot more pressure to drive the same amount of flow through that lymph node. And if you only have the same amount of pressure necessary, then a lot less flow goes through that lymph node. So this is an interesting and quite striking change in flow resistance. And within 72 hours, it's back down to its baseline level. And if you remember from the last slide, the lymph node is still swollen at 72 hours. So we have really different processes going on here. We think this change in flow resistance is related to the rapid swelling of the lymph node, but clearly there's a lot more going on here than meets the eye, and we look forward to learning more about it. So then we wanted to know, well, what part of the node changes to increase the resistance? Does it have to do with that core in the middle of the node? Does it have to do with one of the pathways around the outside? And again, these things are really not possible to measure inside an experiment like I showed you earlier. So we went back to the model, And we started changing it. So you know, when it's a computer model, you can reach in and program a different number in, and you get a simulation. So we tried changing the permeability of this core in the middle here, this T cell cortex, and it didn't make any difference at all. Remember, most of the flow goes around the edge anyway under baseline conditions. So if you change what goes on in the middle, it doesn't really make any difference. Then we changed the subcapsular sinus properties, didn't make any difference. And then we changed the permeability of the medullary sinus, so that collection point where all the flow goes out. And sure enough, that's the only change we could make that would explain this change in flow resistance. So this is where we're looking right now, biologically, is in the medullary sinus to try and figure out what changed down here to change that flow resistance. Now, the good news about the increase in flow resistance is that when you shut down this low resistance pathway around the outside, then more of the flow goes into that core in the middle. So the amount of flow going into the T cells and the B cells triples when you increase that flow resistance. So that's good news, and that makes sense because you want to send more of the information into those cells in the middle. Right, so let's go back to our picture here of the vaccination. So though I think the proper way to think about this is start with the lymph node. So what information does the lymph node need, and what time does it need it? Does it need a bolus of information right away, or does it need a slow, steady delivery of information later on? Well, it turns out nobody really knows, but I think, you know, we'd like to know more about that. But I think that's a way to start thinking about how we might maximize vaccines. So then the next step back is the delivery of the information. So the injection is at the muscle, but the nearest lymph node's a couple of centimeters away and it has to get there somehow. And that's the job of these pumping vessels, which I'll talk about next. But then I think, you know, you got to go back then. How do this process relies on this process. And if we want this process to work well, we need to go via this process to optimize the vaccine. And that's really the way we're thinking about it, is start with the lymph node. Well, let's talk a bit about these collecting vessels here, and this will be sort of the last thing I talk about today. This is what those vessels look like. So this is the lymphatic vessel here. Down here is a blood vessel. And you can see lymphocytes and other immune cells flowing through the lymph vessel. You can see they speed up, they slow down, they go backwards a little bit, but they go mostly forwards. And so this gives you some idea of what those vessels look like and what the flow in them looks like. The velocity of those immune cells going through there is about 1 centimeter per second. So if your arm muscle is 5 centimeters away from the nearest lymph node, then the information will get there within a minute. And we see that all the time when we inject things into mice or humans, that the information gets to the lymph node literally in minutes, right? And so that whole pumping process, I know I talked about it last time at the seminar, but at the previous one, but basically the way these vessels pump is they have these check valves that keep flow moving in the right direction. And then they squeeze, and that's with the active contraction of the muscle cells in the wall or adjacent tissue movement. But basically squeezing plus check valves is how you pump in the lymphatic system. And this I also showed last time, but this is a pumping lymphatic vessel that's been taken out of the body. And I wanted to show you this because you can see the valves here. So there's a valve there and there's a valve here, and you can see when the vessel squeezes, those valves open and close and keep the flow moving in the right direction. So that's kind of the basics of the way the lymphatic system pumps. Now, let's talk time scales, because remember, you want to get the information from the injection point to the nearest lymph node as quickly as possible, you would guess, right? So how do you get that information there? Well, there are cells in the arm that can take up the antigen, and they can crawl. They have the ability to crawl around, much like I showed you that cells crawling around in the lymph node. And so I've had biologists ask me, well, what about cell crawling? Well, the velocity of those cells crawling is about 5 nanometers per minute. A nanometer is 1 billionth of a meter, so a very tiny distance, right? So that's the velocity those cells move at. And remember, I just told you that when a lymphatic cell, when a lymph cell immune cell is moving through a lymphatic vessel, it travels at 1 centimeter per second. So this is going 1 centimeter in 4 years. So this is not a very, cell crawling is not a very good way to get information into a lymph node from the injection site, but that's still unfortunately what a lot of biologists believe. And I have another analogy for you here. So if you imagine this is my lab group. We like to do a hike every summer. So the speed at which they were walking on this hike, if you equate that to the speed of crawling vessels, then the speed of the cells that get caught in the flow of those pumping lymphatic vessels is the equivalent of about the speed of light. So if you're trying to get information from the lymph node, sorry, from the injection site to the lymph node, it's better to travel at a much higher velocity. So that's a great thing about the fluid flow in the lymphatic system. So back to our friend Alexander Glennie. He came up with this aluminum salt as the vaccine adjuvant, and we wanted to know, well, what effect do these adjuvants have on the lymphatic pumping? Well, it turns out that if you look at the way these vessels pump, again with the squeezing and all of that, the maximum diameter of these vessels goes up when you give alum, and the contraction frequency goes down. In other words, this shuts down lymphatic pumping. And you think, well, I mean, we just didn't expect this at all. We thought it would kick up lymphatic pumping, right? So it shuts down lymphatic pumping. And these are the other two kinds of adjuvants I told you earlier, the oil and water emulsion and the TLR activator. So every adjuvant we've ever tried shuts down lymphatic pumping. So let's go back to our cartoon here of vaccination. So step one is to get it taken up into the initial lymphatics. So what effects do adjuvants have there? Well, we don't really know. It's been observed there's a bit of inflammation at the injection site, but that observation is 100 years old, and we need more science behind it. What I've shown you today is that adjuvants actually shut down the pumping of these lymphatic vessels whose job it is to deliver the information to the lymph node. It's not completely shut down, it's just slowed down. But is that a good thing? And is that the mechanism by which adjuvants help you have adaptive immunity to a vaccine? We don't really know. Again, we'd like to start with the lymph node, and from what we found there, it actually speeds the delivery of the information into the T cells and B cells when you give the adjuvant to the lymph node. So there's a lot of interesting stuff here. We're not totally sure what's going on, but we've got a good start on trying to figure some of it out. So where are we headed now? Well, this nice movie you're seeing right now is an image, a full 3D image of the blood vasculature inside a mouse lymph node. Remember, this lymph node is only about 1 millimeter in diameter. They're really small little guys. So these lymph node blood vessels are really interesting. They're responsible for fluid exchange inside the lymph node. So there's exchange between the lymph compartment and the blood compartment inside lymph nodes. A lot of cells get transported across the walls of these vessels. There are changes in lymph node structure with vaccination, as you've seen earlier. There are big changes in lymph nodes with cancer, and we'd like to know what happens to these blood vessels. It's never been imaged, never been measured, and we'd love to know more about that. And finally, what can we do for vaccines? Well, I think what we provide is the possibility of a platform for deciding, providing some rational basis for choosing a particular adjuvant for a particular vaccine. And I think that's really a lot of potential for societal impact in making vaccines better in that regard. So I'll wrap it up now. As you can see, lymphatics, it's mysterious. I've had to admit many times I don't know what's going on in these vessels or in these lymph nodes because there's just not that much work done on it. We like the pumping, and we like the vaccine story, and look forward to pushing that further. And with that, I'll close up with the names of the people and the funding sources that really made this work possible. And I look forward to answering whatever questions you have.
Jimmy, thank you very much for that. Honestly, when I see some of the stuff that you guys do, it blows my mind still when you're talking about how small these nodes, vessels are. So it's so amazing what you guys do, and hopefully, getting this across to people on the front line really helps sort of build people's knowledge behind it as well. Like you say, it's an understudied area.
Yeah, thank you.
It's getting that information out, I think, is really, really important. We've got a couple of questions come in already. So obviously, you know, when we talk to lymphatics, we'll get a lot of lymphatic specialists on there, people who are dealing with dysfunctional lymphatic systems. So Sue Hansard has asked, if a node responds to a vaccine by swelling and changes in flow then occur, increasing an exchange and increasing exchange of information to cells, does this mean that where an individual experienced no nodal swelling, there is a lower response?
Well, hi, Sue. Thanks for joining. Not necessarily. We know that there's a lot of individual variability in response to vaccines. I got mildly ill after my first jab for COVID. It could be that that adaptive immunity is happening just fine without a huge lymph node response in terms of swelling and fluid backup and all that. So that's something we'd like to know more about. I think there are good reasons for lymph node swelling, and we've explored that in some of our other research, that it does help you develop the right kinds of cells that provide, first of all, robust immune response right away, and second of all, lasting. So those memory cells that keep the memory around of the antigen really need that it helps to have the swelling. But I wouldn't worry if your lymph nodes don't swell. Chances are the vaccine's working fine.
Perfect. And just to say, reminder to anybody else that has joined us during the webinar, saying that the Q&A function is at the bottom of the screen. If you put your questions in the Q&A, we'll get those posed to Jimmy. Obviously, we're talking about lymphatics, Jimmy. And we've been talking about, obviously, you've been looking at healthy lymphatics. There's going to be a lot of people out there that are dealing with dysfunctional lymphatic systems or the lymphatic disease. When we're talking about vaccinations and things like that, which is very apt at the moment, how would it affect, or is there any effects or differences with dysfunctional systems where there's limited flow, no flow, or no uptake?
Yeah, well, it's we're still gathering the information, and my strong feeling is that if you have something that stops that information from being delivered to the lymph node, I don't think the vaccine is going to be as effective. So there have been experiments with animals where they inject directly into the lymph node, and it works great, right? But it's difficult to do that in people. But so that's why we rely on the lymphatics to deliver it there. So I would say avoid an injection site that's near a dysfunctional lymphatic bed. But I'm not a doctor, and I would trust what your doctor says in that regard. I think it's something that we need to know more about, and obviously we want vaccines to work in everyone, especially people with lymphatic dysfunction, because their immune function is going to be compromised as well.
Yeah, I suppose that's the other aspect, isn't it? Is when your lymphatic system, if it's dysfunctional, then that immune capacity is potentially compromised. It's like, is there any way around, or is it just like people have been doing? They've been sort of shielding people like that, or is there any way that is it just give them a vaccination anyway and just kind of hope it uptakes and hope that those cells respond and take on the vaccine?
Yeah, you just have to hope for the best. I mean, we know vaccines don't work in all healthy people, for that matter, but that's why it's important for everyone to get vaccinated, because then you protect those in whom the vaccine didn't work, and so that's an epidemiological and public health issue as much as a lymphatic issue, really.
Yeah, and I think I'm pretty confident in saying that with particular conditions, diseases like lipedema and lymphedema, where there is dysfunctional systems for whatever reason, whether it's from cancer surgeries or whether it's people that have been born with it. I know that there'll be somebody on here that will probably hopefully back me up on saying this, that if you're trying to avoid, if it's a unilateral, you're trying to avoid injection in that side, you're trying to go on the healthy side. I know there's obviously the risk if you're injecting into edematous tissues or lymphatic dysfunctions, you're breaking the skin, there's risk of infections. So obviously there's a lot of things to to consider when it's that kind of patient that you could be presented with. So I mean, on the side of things, do you know of any kind of medications or vaccines that have a negative impact on the lymphatics?
Yeah, I guess you'd have to qualify negative effects. So, you know, there are definitely substances that are produced naturally by the body that slow down lymphatic pumping, and nitric oxide being one, which is a quite famous little molecule. It's you know, and there are medicines that encourage the production of nitric oxide, for example, and so those have been shown to slow down lymphatic pumping for sure. So there are ways of modulating lymphatic pumping if we know what the goal is, right? So do we want to speed up the pumping? Do we want to slow it down? And I don't think we know, even for a vaccine. You know, do we want to slow down the delivery so that the node gets a little bit of information at a time, or do we want to hit it with a bolus of information and then sustain that, or you know what? And that's just, you know, we just don't know. So you know, yes, there are ways of modulating lymphatic pumping, and those substances are pretty well known. But you know, to what end is the real question.
Yeah. I suppose as well, like I know from an acute infection perspective, we don't do any manual work. So, you know, lymphatic drainage, massage, you know, most treatments when we're looking at injuries or, you know, lymphatic drainage, whichever, if someone's preventing with acute infection, we don't touch them. Is that the same for a vaccine? Because surely if you'd want to get it to the lymph nodes quicker, you want to get that around the body quicker, could you do a vaccine and then massage?
I've heard of that being done. I don't know that there's much proof that it helps. I would say it probably couldn't hurt. But yeah, it's
the whole idea with like an infection is the body wants to contain an infection. As soon as you open up the lymphatic system, it's able to go free. But there's always been that discussion of vaccine. If I don't know.
Yeah. Well, the antigen, the version of the antigen that's in the vaccine should not make you ill, right? So there's certainly no danger. I don't think there's much danger in doing that, but I don't know if there's much science behind it.
And just explain. I'm just so I'm right. I'm hoping I'm not the only one. I'm not the only person thinking this. The antigen is like part of the strain of the virus.
That's right. It's enough information about the virus to alert your body to look out for it if you ever see this.
And it's building up those memory cells so that if the virus then enters the body, it's already got that antigen receptor there.
Yeah. So the memory cells are these lymphocytes that we talked about, where they have ways of recognizing if that antigen ever shows up, though, if you ever get infected by the thing you've been vaccinated against. They go there, they recognize it, and they call their friends in. They call in the army, and it gets destroyed.
Uh-huh. So I'm not sure how sort of well versed you are with the COVID vaccination, Jimmy. We've had a question of, do you know if there's a greater chance of cellulitis in the lower leg after a COVID-19 vaccinations for patients with chronic edema?
I don't know. Sorry. Yeah.
No, that's fine.
It's a good question. I hope someone knows the answer, because that we want to do right by the people suffering from these conditions, for sure.
For whoever's asked that question, it might just be one to throw out there on LinkedIn and see if somebody knows. I'm sure that LinkedIn or Twitter, there'll be somebody that may know the answer to that question. So apologies, can't get that one answered for you. Jill Drew Griffiths has asked, what are your thoughts on manual lymphatic drainage for long COVID symptoms in patients? Again, if you have any experience with that or know anything with that.
Well, I mean, I know someone that's had long COVID, and I'll tell you, it's terrible. And I don't know if they've tried that. I don't know of any studies of it. I mean, anything we could do to help those people, we should be doing it. I would put in the category of probably doesn't hurt and could help. So, whatever we can do to help them, obviously. And actually, I just realized I should advance to the Q&A slide, I guess, at this point. Sorry about that.
No. I think just to add on to that, I think from Jill, from your perspective, I'd be, probably speaking to the MLD groups or some of the people that are working in that area. They'll
Yeah.
hopefully, I'm sure they'll probably know more for the application of that technique. So, Jimmy, just with the obviously with the lymphatic system, you mentioned the toll-like receptor. Again, I hope I'm not the only person that's thinking this, but I'm just going to take the opportunity. Toll-like receptors, what are they?
Yeah, so they're very, very powerful receptors on some cells in the body that instigate an immune reaction. So they typically are relevant for responding to bacteria. So it's just a really powerful way of triggering the immune system. So, and here's a good lesson of the shaky relevance of studies in mice. So toll-like receptors sort of came up in mice, and they worked great in mice. And in humans, some of them are toxic. So we've had to be very, very careful about how we apply toll-like receptors. So there are not many vaccines that use toll-like receptor agonists as the adjuvant.
I can only imagine just the amount of work that goes into creating vaccines. And
it's a lot of guesswork, honestly.
That's it.
Yeah. There's not a lot of quantitative knowledge base on which to sort of decide which adjuvant to use for a vaccine. So that's where we'd like to help.
Yeah. And with we're talking about sort of the lymph flow into the node. Have you done any experiments or any measurements on dysfunctional nodes to see how they're responding?
Not beyond what I showed today. I mean, it's, we'd love to get into looking at vaccinated nodes. The internal structure, how the blood vessel structure changes. We haven't been able to do that yet. So that one vessel structure that I showed you, we've imaged, I don't know, 5 or 10 lymph nodes, and they're all healthy. So we've not had we've applied for funding, but not been successful in trying to image the blood vessel network inside vaccinated lymph nodes. We've also tried unsuccessfully to get funding for to look at the blood vessel structure inside cancerous lymph nodes. So where metastatic cells have come. I think there's a lot of really interesting work that could be done there, and hopefully we'll land some funding soon to do that.
So I think that would be really interesting. I'm thinking as well, like following from like cancer surgeries when a certain amount of nodes are taken out, or even looking at the impact of lymph node flow with cancer patients pre-post. I mean, do you think there will ever be a way that we could potentially measure lymph flow in the node on live humans, where it's maybe injecting something and looking at something under a microscope under a live subject? Do you think that is it beyond the realms of possibility, or is
it would be very difficult? So the flow rates through lymph nodes are tiny. I mean, they're on the order of sort of hundreds of microliters per hour.
Okay, so you could be there a while.
Yeah, it could be there a while. It's the good thing about the lymphatic system is it's pretty easy to get substances in there to label the fluid that's going through. But the problem is the way you see that dye moving. For example, in that lymph node movie that I showed you with the red dye coming in, that dye is diffusing about as fast as the fluid is moving. So it's it's sort of hard to get a good fluid velocity measurement out of that. But at least you can see where our stuff is going, and that. But again, these structures are tiny. So yeah, a lot of good challenges for our colleagues that work in magnetic resonance imaging and ultrasound. As they're making some good headway with that at Imperial as well.
Oh, interesting.
Yeah.
I mean, talking about lymph nodes, Jimmy, I know you're working on another project with lymph nodes. How are you getting on with that? I think we mentioned this last time. I think, how are you getting on with that project?
Yeah, so we actually got a nice grant from NIHR late last year to work on a lymph node implant. So the idea with that is it would go in at the time of the breast cancer surgery, replace the lymph node, reconnect the lymphatic vessels, and it's not intended to replace any immune function. Really, it's just trying to keep fluid moving and hopefully prevent patients from getting breast cancer related lymphedema. And of course, we'd like to show success there. It's a massive problem, as you know, but then to also make that available to other cancer patients as well. So that's it's an exciting new project for us, and look forward to getting it out there where it can really help some patients.
Very exciting. So just as a concept, then it would be, you know, when patients have an axillary clearance, is that as they take out one node,
that's right, Don. Yeah,
they're putting in this transplant that, like you say, doesn't help with the immune system, but helps with the flow to prevent that backup.
Yeah. So literally, the surgeon could sew it in 20 seconds, and you know, then they close them up and they're done, right? So it doesn't interrupt the surgical procedure really that much at all, and hopefully it prevents that, you know, horrible condition from forming.
I look forward to hearing how that goes. That's very interesting. Very interesting.
Thank you. We're very excited about it.
I know. I'm hoping there's a few people going to be watching this thinking, "Oh, that sounds really interesting." So, look, there's no more questions coming in. I've run out of questions my end. I've said that, and there's somebody who's popped up a question. Let me just see what's come up. Will you look at the effects on the donor sites from where a node is transplanted?
So that's not something we've looked at. So I think they're talking about autologous lymph node transfer.
Yes.
Right.
Essentially,
so, yeah. So that's where they take a lymph node, say from the groin, and move it up to the axilla. So that's actually a procedure that we'd like to replace with our implant. But you know, for the time being, it's you know, hopefully it's doing some good out there. You know, it's you know, removing a lymph node from the groin is obviously not something to be taken lightly, but we've never looked at any of the flow patterns around that really.
So I mean, to clarify with Stu, the transplant that Jimmy's making, it's a synthetic implant as such.
Yeah, that's right. So we wouldn't need a donor lymph node. Yeah.
Say again, sorry?
We would not need the donor lymph node. It would just go in. It's a piece of plastic with a drug coated on it. So.
Yeah, I mean, I've been fortunate enough to see one of those things. They are tiny, but quite incredible, really. So I'm looking forward to seeing that. Right, Jimmy, we'll wrap it up there. If anybody does have any questions afterwards, then please feel free to submit. Of course, we're going to say we'll send out the recording so you can watch it all back, take some any additional notes if you've missed anything, and then submit any questions afterwards. Everybody who joined us on the live session this evening, thank you very much for your time and your participation. Jimmy, once again, thank you very much for your expertise. Absolutely fascinating. It's just another level of what you guys are doing, and it's honestly brilliant. And thank you again for joining us and sharing all the work that you do.
It's been my pleasure. It's a real honor, and thank you, Dom and Kate, from PhysiQuip, and you know, all the best to you and the great work you're doing as well.
No, thank you very much. So for those people who've been on, you'll receive an email following this with the survey. Again, if you can just take a minute just to fill in this survey, just let us know what your feedback is and any future things you would like to see, either from us at PhysiQuip or something from Jimmy. Send us your ideas and we'll see if we can facilitate that. We'll also, like I say, we'll send the recording so you can watch it back. In terms of upcoming webinars, Jimmy, if you could just flick onto the next slide. I think we've got some upcoming webinars for people. So talking about COVID quite a bit, we're putting on another webinar, which is managing vocational rehabilitation for a long COVID patient. So if you're interested on that, you can find out on the 14th of June, 2 p.m. Again, if you can't make the live session, register your details and you can get sent the recording, and please send that to any of your colleagues you feel may be interested in that as well. Otherwise, everybody, thank you very much for your time. Wrap it up and we'll have a good evening. Enjoy the weather. Everybody keep hydrated. Isn't it? It's very warm at the moment. So yeah, Jimmy, once again, thank you very much. Take care.
Thank you, Dom. Bye-bye.